personally think we have all the tools necessary to really have breakthrough in curing incurable diseases and finding drugs and things like that. I think we have all the tools. I just think that whether we are approaching it right or not, I don't know. I don't know actually. Maybe we are. Maybe we'll start to see some big wins coming in. I really would like to see that. But as I said, like my approach is always going to be multidisciplinary. I need everybody on the table around me, at least 5 disciplined people, and then start arguing with each other and fighting with each other. I think that's when things come out.
Hey everybody, I'm Costas.
And I'm Lester, and welcome to another episode of What's with RWE. And of course, we have another fantastic guest today, actually our second guest in a row that is not Canadian. So we're actually on a roll in terms of international guests. We have the wonderful Shahed Iqbal on today. He is the Executive Director, Molecular Epidemiology, Clinical Data Science, and Real-World Evidence at Gilead. But before we jump right in, so just so everyone knows today that Shahed is on, all his comments reflect his own personal opinion and do not reflect the opinion of his employer, Gilead. So welcome to the podcast, Shahed.
Thank you, Lester. Thank you, Costas, for inviting me. Happy to chat over real-world evidence.
Excellent. It seems like you have tons of sunshine in your background. Where are you based again? You're in California, right?
Yeah, we're in the Bay Area, so my workplace is in Foster City, but I'm on the East Bay.
And you must be super warm right now, as opposed to us. We're I don't know. How about you, Costas? Are you negative yet in Toronto?
No, lucky today we just went above the 0 degree mark, and we're just have fog and drizzle out there. So it would be a nice day to be on the West Coast.
Yeah, it doesn't get very cold here, but it does get a little cool on the cool side. So it's starting to get there, but it's still very nice.
Yeah, exactly. It's winter time. So thanks a lot again, Shahed, for jumping on. And today we're doing something slightly different. We actually haven't explored real world evidence in this angle yet on the podcast. There's really the use of real world data in research and development applications. So Shahed's an expert in this area, so very much appreciate him taking the time to join us today. So maybe Shahed, if you could take a little bit of time to just maybe explain to everyone like why did you get into this specific space, real world data? Why is it important to you, and how did you get into it? Because it is somewhat of an esoteric place to go to, because I assume most kids don't necessarily want to be an epidemiologist when they grow up. Maybe more of a athlete or some sort of like rock superstar, right?
So yeah, it's kind of like a long convoluted path to where I am right now, and it's pretty consequential, I must say. I have a background in medicine, but I never really wanted to go into clinical practice and wanted to work in public health. So I was trained as an epidemiologist, got my doctoral degree, and then I went to the Centers for Disease Control and Prevention, CDC, at the US as an Epidemic Intelligence Service Officer, an EIS officer, just basic shoe leather epidemiology. So initially my focus was more on environmental health, asthma, lead poisoning, healthy homes, air pollution, things of that sort. But later I transitioned into a position in vaccine safety, which is very important. And while at vaccine safety, I guess during this time, I was getting exposed to a lot of large-scale patient-level data sets that were becoming available at the time. So what we call the claims databases, the hospitalization databases, the ARC databases that were coming out of the HCUP databases, and market scan, all those things. So I got interested in exploring them more. And we also had access to, from as a CDC, we had access to lots of vaccine safety databases with Kaiser and other healthcare providers. And these large-scale databases, we saw the potential that how it can really replace the prospective studies we do. Anyway, so what happened in the last 15, 20 years is like a lot of these databases became available. And at that time, I transitioned into drug safety into industry. And during that time, I saw that they are not only becoming available with large number of patients, but they're also available coming up with a lot of what do we say, genomic or molecular data attached to them, which was fascinating. That prior to that in industry, we used real world evidence extensively in drug safety, post marketing monitoring of safety and effectiveness. We helped understand the natural history of disease, the patient stratifications, the heterogeneous disease subgroups, and all this stuff. And we also helped the clinical development and strategies with treatment patterns, understanding those kind of things, and management of the patients, screening of the patients. But with this new kind of data that were coming out, the modalities, the molecular, and also the whole healthcare innovation shifted towards precision medicine. So, and I thought like, this was a good opportunity to actually mine or explore these databases and actually use the epidemiologic skills along with advanced analytics to get some to really help the earlier stages, the translational stages of things that we can actually help a lot of things, translational and reverse translational work we can do. And that was my I was put that brought me to Gilead, and we started this function. And then my work right now is more, I would say, translational and reverse translational using molecular, genomic, transcriptomic data. And just to mention in this regard that in the last 10, 15 years, a lot of things happened. Not only that we had a lot of data, a lot of modalities of data like genomic, transcriptomic, proteomic, but we also had the technological advancement, the cloud computing, the advanced analytics to actually efficiently analyze and mine these data sets. So there was a convergence, and there was also a favorable environment in terms of investments from private equities and VCs, and also from regulators in how they will accept this kind of data. So a lot of convergence there that actually brought real world evidence into one of the core, becoming a core functionality within drug discovery and development.
Wow, it certainly does seem like there was just a lot of events coming together to take us where we are
Yes.
today. But maybe let's take a step back. We have a very diverse audience, and let's talk a little bit about what does it mean when we say translational medicine, translational R&D, and reverse translational, and then let's talk about why real world evidence is so important in the way that those two things are done today.
So one aspect is like when we say translational, the general sense is like we are looking at the translation of evidence from bench to bedside, meaning what we are having in bench research, are those things holding up in our patients or not. So
Yeah.
basically, the in this is biology, computational biology, or even like discovery. We piece together a lot of information that are in vitro, in vivo analysis and things like that to understand the mechanism of actions of the molecule and sometimes the disease itself. And we want to see if we see the same level of if that holds true in the patients or not, because we have now have the patient data. And so we can, in that way, we can validate some of the assumptions during our discovery process.
Mhm.
We can better understand the mechanisms of actions, and for some drugs, the mechanisms of resistance, like which genes or which variants or which mutations are actually contributing to the resistance, things like that. So we can inform that early space a lot. And then on the reverse side of it is like during our analysis of the patient level data, we might find something interesting that this could be a pathway that should be explored more in for resistance, or this should be a pathway that could be explored more for combination strategies with other molecules, or for discovery, pure discovery sometimes. Um, Not to like so that there is like a huge utility there. But I will also like temper the expectation a little bit that it's not always conclusive. It's a piece of evidence like we have pieced together many pieces of evidence before we go forward or refute something. So it's going to be one piece of evidence that will have different degree of confidence in it depending on the data and the results, the findings.
Right. So it's essentially it allows you to almost crystal ball and pick assets that are more aligned, let's say, to what you're targeting. So do these assets behave in the way that you expect them to, or can you explain how they are delivering their impact on patients, or in vivo, even in some cases, depending on how early it is, that to understand that mechanism. So this is the bridge to the real world, to what we
Yes.
think it may do in patients eventually.
Yeah, definitely. It's a bridge. But as I just said, like in an ideal world, it would be crystal clear. But we have
Yeah,
real world data. We go into niche indications, and when our patient population dwindles, that is not they do not have enough. They're not powered to do discovery, and there's not enough power to do stratified analysis, and also there's a like it's wrought with the the lackings of real world data, like the same missing
yeah,
values, lack of standardizations, inconsistent results, and things like that. So there's a lot of things we have to clear up before we can go there. But yes, but it can really aid, as I said, can add a significant piece of evidence to our understanding of the whole biology and the mechanisms of actions and how it might behave. So there's a huge potential that we are still exploring. It's still in a very early phase that we cannot say we are doing it on a regular basis, but we are doing it as needed sometimes.
Yeah,
right.
that's really that's incredibly interesting and fascinating. But just in case there are billions of people watching right now from across the world, Shahed, for those of us who are not scientists and in the know as you are with real world data and this translational and reverse translational, like, is it possible? Can you give an example? And you could just make up an example that is not something that that you're currently doing, but just something that you think potentially could happen.
Right. I mean, I could give a very generic example, like, that everybody's kind of like trying to understand and things like that. Like one of the biggest advancement that we had in oncology was, the discovery of anti-PD-1 drugs and everything, right? Like pembrolizumab or nivolumab, but
Mhm.
only 25 to 30% patient actually respond. And there are actually lots of early and late resistance to these drugs, right? So one thing that we can look at is like, okay, why are some people responding? Why are some people not responding? What is in their molecular profile that is different? So that you can see that if we can identify some of the pathways, some of the genes, or some of the mutations of interest, we can always focus that whether what can we do with that information. That's outside of my wheelhouse, but that can go into some research and they can start thinking about it. There are ways to take it forward with that information. So that's an example of how we can use it, the molecular data, to aid into some understanding and in the discovery process, if I may say.
Got it. Got it. No, thanks a lot for that example. And you spoke a little bit about this. You glossed over it, but I'm sure it's one of the banes of your your working day is issues with real world data. So you mentioned a couple of things already. Maybe just, if you could speak a little bit to the challenges of working with real world data. We understand it's not the panacea that can solve all issues,
Right.
but it offers a light into a specific path that you can potentially take. But I know, I assume working with it is, of course, there are challenges. So maybe you can speak a little bit about that, Shahed.
So yeah, I mean, like, there are two parts to it that we're talking about. One is the molecular side that is coming from the labs and things like that. Other is the clinical part. Let me talk about the clinical side first, because that's been the basic foundation of our understanding of real world data, coming from the claims, coming from the registries or EMR. Basically, claims for EMR, the first thing I would go into a very root cause of it. I mean, first of all, we have to understand that these data that are collected, That's not because, not with the purpose of research. There's some clinical management or billing. So there are different perspectives. So from the get go, it's not ideal in some areas. So we have to do a lot of kind of thinking as to how to approach the data. The second thing is like it may or may not have all the variables we need. And if they even have it, it may or may not have all the variables in the completeness of the data. So the completeness, the correctness of the data, and the consistencies of the data. A lot of the times we get electronic medical records from multiple different hospitals. The practices may be different. So last 15, 20 years, there has been a lot of effort from the community to standardize these data models and to bring us closer to what we call more pristine, more regulatory grade data. And there are ways and methods to do that, and also methods to kind of like combat some of the limitations of the data, but they remain. They remain, and especially when you go into a niche area, I keep bringing it to the thing to oncology, or even like if you look at the population genetic data, UK Biobank, you go into like they're never powered enough for discovery, and you may start with 10,000 patients, you may end up with 100 in your analysis set. So that's the level of attrition we are talking about. So that poses a challenge. On the molecular side, we get data from different. If we're looking at clinical, if we're getting it from the EMR, they are those molecular testing. It has been they have been done on different platforms, assays, and things like that, and they have their differences. How do we take into account? And if they're coming from one single lab, then can we just add to the other lab, and how do we harmonize the data? I mean, TCGA has some harmonization methods, and but we have to harmonize it at a level. And there's also data proprietary data, so some people we cannot do it for ethical reasons or HIPAA reasons and things like that. So lots of challenges, and the last piece of it is like when you add the molecular, it becomes like very expensive. So the scale that we need for large scale discovery is becomes like really unattainable because of the cost. Sometimes it's cost prohibitive to increase the number of samples you need or number of patients with molecular data.
Got it. Got it. Yeah. Thanks a lot for outlining a lot of these challenges. And what do you think is to you the maybe two questions in one? Which one do you would if you had a magic wand, if you could fix today, would just make your life, I don't know, 10x simpler? And what's the one that you think is actually the most pragmatic actually to solve? That's simplest to solve.
Um, the simplest to solve is um, when from because of my work is more focused on clinical genomic data. I can talk about that. I would like to solve like the issue of how we can merge data sets together to increase the sample
Mm.
sizes. That was number one. And the other one is everybody wants to solve is the data quality. And that's basically those are the two. And of course, you asked for one, but I'm giving you three. The cost would be nice to have like to show to if it comes down, we can really achieve a lot of wins with this data, I believe.
Yeah. I think those are bang on, probably the best. I don't think there's anyone there that's a cheap, simple, easy fix,
Yes,
and maybe there isn't any. But I think all of those are not only important to the way that we do work today, that we utilize that data, but I think it's also, if we start sort of future casting a little bit and think about, you know, the application of AI into discovery,
right,
and that early development phase, it is so important, it's so dependent, I should say, on the quality of the data and the volume. I mean, you need large data sets to really be to train models and to be able to
right.
validate them. But you know, is that, I ask, maybe kind of knowing the answer a little bit, but is that starting to already, the use of AI starting to already penetrate into the work that you do now day to day, or is that still sort of on the sidelines waiting for it to get better quality data? where is AI coming in this data world?
Um, yeah, AI is coming whether we invite AI or not. AI is here. So I'm cautiously optimistic about AI. I think it has huge potential. But what I think one of the other challenges that's not typically discussed is like when we are bringing in, just imagine, see, I have like three different modalities of data. See, I have genomic data, transcriptomic data, proteomic data,
Mhm,
and maybe single cell data. Then I have the clinical or phenomic data.
mhm.
The main question is comes like, who owns this paradigm? Whose domain is this, right? You can think like it can belong to a computational biologist. It can belong to a
Yep.
bioinformatician. It can belong to an epidemiologist. It can belong to like an overall data scientist. And I think that also is something we need to sort out. And the answer is to
Mm.
me is like it's not one person. It has to be a multidisciplinary approach. I actually advocate for a new breed of scientists, like which with understanding of every single area a bit and expertise in one area. It's happening, but not happening at scale yet. So, but we are starting to think about AI and use AI. I mean, machine learning has like we have used, but AI is more recently. When we say AI, we refer to LLMs and things like that. We're also thinking how to incorporate, and there are we cannot just wait for things to happen. We have to keep on doing things, and that might lead to a different path. I think that's the approach. Like let's do piecemeal wise and see how useful that is, and then we can scale it up. I think that's what my approach would be.
A little bit hype right now, but whether we like it or not, we need to build that future. I think is the takeaway message there.
Yes.
Yeah, and actually, I absolutely agree with you on the kind of like you need a multidisciplinary approach, but you also need some almost like a different way of thinking because it actually throughout most of I don't know last 2 3 decades when you go through academia and go through graduate school usually what happens from undergrad to graduate school right is like your focus actually narrows right
Right.
it narrows more and more and more and more right until the point like
Exactly.
you're like the expert at this like one super like
Yeah.
esoteric thing right so is you're almost suggesting we need to re-expand in order to really use this
Or
great so go
yeah.
ahead.
No, no. Sorry. I was going to say like, or we need to acknowledge that we have boundaries of our knowledge, and at one point, I need to seek help from the next person, right? If I'm
Yeah.
designing a study, I need to understand the biology. And when I run out of my biology, I need to get a biologist to tell me like if I'm designing something that's biologically plausible or makes me sense or not, or a clinician to say that hey, yeah, clinically important or not. A statistician could come and limit the methods you're trying to do, which are they're good or not, and then the data scientist can say, hey, we can make it do it more efficiently or not. So there are lots of components. I think but I have seen like one person doing everything, right? They would publish paper. I would review papers that had 50 different graphs, and at the end, like I mean, if I ask so what, there's really no answer, and a lot of the times that are contrary to the biology that we understand, right? Not that it cannot be, but
Right.
I'm saying that we need multidisciplinary teams. And I one thing I say that whenever we are talking about discovery translation, it's a biological problem. We cannot just solve a biological problem with an engineering approach. We really need to think about that it's important. We have to have that engineering angle to it, but that cannot solve it. We need the biological approach totally integrated.
Well, you're a real
Yep.
renaissance man. It's a very it no it's completely very apropos because there's so many things in the because unless you've gone through grad school right it's for people that haven't gone through grad school it's one of those situations where when you're amongst a whole bunch of people that are in some ways are trying to pursue something that's greater than themselves but they actually don't like to ask for help I do notice that a lot of people that have gone through that system don't like to ask for help, right? And because it seemed as if you ask for help, that there's a deficiency in your intellect when you do that, right? So you almost have to
Right.
change it when you in order to actually leverage this new technology.
Definitely. And this technology and the methods are fascinating. Sometimes it's I don't understand it, so I go to my teammates actually or to someone else, like explain it. Why should I use this over my traditional methods? What is it
Yeah.
giving me? What's the advantage? I mean, like I can still use a paper map, but like a map on the my phone is more convenient. But to what, like what's the benefit cost ratio, right? So I would like to see that this is what we are finding. What are the big wins we have? I mean, on a like a major large scale, if you look at it, AI for efficiency purposes, I think AI will be super helpful, excellent. I mean, but in terms of novelty, innovation in biological space, we have to be careful. We have to bring in people from different disciplines. Otherwise, I'm doubtful that we'd have big wins.
Yeah, but I think bringing those other experts in that typically don't sit that early, or
Right.
bringing in analogous experiences is also an innovative approach to what has the way that we have done translational medicine early development for the last 40, 50 years probably, at a
Right.
very trial and error approach. And we've kind of hit that plateau where you can only do so much. You can find many assets, but then how do you decide to take one forward with limited funds? So, anything that we can beg, borrow, or steal from another discipline that helps to innovate, I think that's going to be a fascinating way to just unlock the potential.
It's easier said than done, right? So it's very difficult to do that. We can
Absolutely.
theorize everything we want in a utopic world, but it won't happen that easily. I think there needs to be commitments. There needs to be organizational readiness in kind of bringing creating that space. We always talk about breaking silos, and we really need to mean it, act on it. Like really breaking silos, like
Yeah.
and it would be difficult, but because there are so many things. But I personally think we have all the tools. I
Mm.
think personally think we have all the tools necessary to really have breakthrough in curing incurable diseases and finding drugs and things like that. I think we have all the tools. I just think that whether we are approaching it right or not, I don't know. I don't know actually. Maybe we are. Maybe we'll start to see some big wins coming in. I really would like to see that. But as I said, like my approach is always going to be multidisciplinary. I need everybody on the table around me, at least five disciplined people, and then start arguing with each other and fighting with each other. I think that's when good things come out.
I like it. It'd be a good table to be around, I think.
Yeah. As long as it's done with respect, right? That's the whole thing. As long as
Yes,
you
yes.
like it's
Yeah.
respect, it's
You
you can disagree.
yes. You have to have trust in your stakeholders, your partners, right? That you all mean have a common goal and you all mean well. Then everybody will have to be at least say their thoughts and have their part of the responsibilities. And people usually do actually. People are more mature than we give them credit for. More professional, more professional, more professional. Yes.
Absolutely, absolutely. I know you're running late potentially for another meeting, so I don't want to take up too much for your time. I have two more questions, both around still around the AI and data side of things. But another AI question, just because since we went down that track, this is much more like a theoretical thing that I never understood how far we are from achieving this given with all the molecular tools that you said that we have now to probe biology and all the technologies to analyze the data itself, like the vastest amounts of data, and then now you have AI to actually make the links, actually find the patterns. Like how far do you think we are to understanding almost like the entire biology of a person? And what I mean by that is, you know, it, a, a person's biology is almost like made up of a sequence of events that are the so-called these biochemical pathways. Like how far are we from understanding almost like how everything is mapped together? Basically, it's like the map of the human biology. Like how? What's your sense of that?
My sense of that, we need to redo that now that we have all the tools, we have large numbers. To be honest, I mean, like, we had that's my sense. And you're asking me about AI. I'm not an expert on AI. I really rely on my AI colleagues and other teammates who are experts in AI. But it's revisiting the biology is not a bad idea. I mean, like a lot of biotechs are doing that, but it doesn't. The question is like, you can only get resources if you can monetize that. Can you really monetize that? You can, is it too big for an academic endeavor? Is it, should it be like a government approach from NIH or somebody like, NSF? For biotechs, they cannot handle these big things. Big industry, should do there be a consortium, an academic industry partnership, public, and bringing in the even the government? I think that could be an approach. Like, hey, let's look at everything. Let's look at not everything. Let's look at a few things that we already know and see if we can map them better or not. I think there are some efforts there happening, and to tackle it holistically, our biology is much more complicated than we just think of. A few pathways is really amazing. I mean, like, after all these years, I'm still amazed of how complex it is and how a single cellular function is so diverse and multidimensional. So yeah, but we need to tackle that. Someone needs to tackle that, and but who's going to do it is probably the big question.
Yes, because you really have a lot of almost, I would say almost like home scientists that are endeavoring in this. And what I mean by that is I don't know if you're familiar with a person named, I think his name is Brian Johnson. I think I forgot how he made his billions of dollars, but he's trying to live forever, and he just does like little experiments on himself where he tries like different things, right? And he like publishes all the data like that so that everyone's publicly visible.
Yeah, I mean, like, I know of him. I mean, a lot of people are interested in longevity. I think there's a lot of things there. I personally don't think that is the primary problem with, of humankind we have now. We have other problems. Maybe we should focus on the at the chronic disease level, at the infectious disease level. We could not. We saw how everything kind of like fell apart in the beginning with just one virus, right? So maybe there are other things we should be prepared for. I mean, like, but sharing the data is important, but also with the privacy laws, it becomes becoming increasingly difficult, and with the genomic data sharing, which is needed, is also very difficult, and some most of the time for right reasons, I believe. So we have to work around these limitations and these like barriers. But yeah, I mean, like, public data repository, I think it has grown like significantly in the last 5-10 years, so it will continue to grow. And as Costas was saying, the models will come into play more and more as it grows, right?
How has um, FDA responded to a lot of this new approaches with data? I mean, I know they're more open to real world evidence, but when you're using that, as just one piece, and you're building models from it, rather than, hey, listen, we've got an assay, this is what the assay shows, we have a biological equivalent, it all falls in line and makes sense, rather than this is what it shows and it seems to map to the real world. There's a lot of white space in between. We're not quite sure how it does, but there seems to be, at least from here to here, it seems to map pretty well. How open are they when there's that kind of uncertainty, when you're going from the early to the big picture without a full sense of what's happening in between?
So I have to admit that last 5-6 years I have not been working on the regulatory side of things a lot. So, but previously, in the last, as I say, 15 years, it has the discussions we had around real world evidence and how to incorporate, I think it just increased. I think there's a lot of thinking going in there. And but the gain is incremental over the years. I think slowly but surely we're heading into that direction, and a lot will depend on the data quality increase, right? And FDA,
Right.
if they have any reservation that is around that, and also, but recently there are at least 4 or 5 guidelines, and one including how to do, I in other, if I may say, like pragmatic trials, like using real world
Hmm.
patients in real world settings, and including them in trial, their information to the trials and things like that. So there is progress, and I'm hopeful, but I think, like, it is growing. The interest and the discussions and possibilities are growing. So, but as I said, like in recent times, I've not been very up to date on what's happening, so I should not comment any further.
Fair enough. Fair enough.
Yeah, and I don't want to take up too much more time, so maybe last but not least, this question it kind of always pops up. It's around the concept of diversity of data, and you can take it kind of whatever way you would like, because a lot of people, when they think of of data, it's a lot of it's around, let's say, racial diversity, but I think that's not the only type of diversity, right? So maybe you can speak on the importance of, is it really important to have diverse data sets, and is it sometimes like overplayed that we need this type of diversity, given the fact that the vast majority of human beings are very similar, at least genetically, right? Genetically, we're very 99.9 whatever percentage, right? It's tiny, tiny differences between people. So maybe you can comment around the diversity of data and the importance in R&D.
Uh, Once again, I have very limited knowledge in that area, but I would say one thing, that the genomic diversity is, we need that diversity data for many different reasons. And you may think we're 99.9% similar, but we're only 2% away from becoming monkeys, right? So, like, that's that. So even 0.1% is sometimes a lot. So uh, we need that, but diversity in every setting is actually needed, and we don't have that. And we do have racial or economically disadvantaged groups less represented in many data sets. So when you're designing a drug or like a study, they're not always captured, and that's very unfortunate, to be honest. I think like we have discussed more about diversity than actually we have tried to solve it. There is lots of efforts, and I think it should continue till we get the appropriate representation. And because in terms of clinical management, in terms of resourcing, that's obviously they are top of the list, that where you need it. Even in discovery and drug development, I think it's becoming increasingly important, and especially precision medicine. I think there is huge lack of data from some of the population subgroups, that we really need to get those data available or measure them. So that's the, like a what do you call it, 10,000 foot view, or like a, but not, I cannot tell you about the what's happening at the granular level at this point.
Understood. Actually, out of
Yeah.
curiosity, from that specific space, because even there's diversity within the U.S. in terms of like racial diversity and different subgroups, subpopulations in the U.S., but it's also global, right? Like
Yes.
we're trying to cure disease for everyone. I mean, I assume that's why you're in this space, and of course, there's countries that just don't have as much of this type of data set. I mean,
Right.
than like to say the U.S. If maybe you can walk people through, just like you said in the past, where you gave an example, like why is diversity important? Like why do you need that representation in the data? Like how does that affect sort of the R&D process and affect sort of the drug that gets developed at the end of the day?
So I mean, like, once again, I probably am not the best person to talk to that, because diversity is such an elaborate issue, and you're talking about a very all through the R&D life cycle. There are different points that we need that diversity to come in. But from, as I said, from the genomic perspective, if we are developing a drug or a target evaluating that is driven by genomics, we need to understand whether that would be applicable to the population that are not represented. Right. That's basic. That whether that will be, if we are talking about some variant that is very predominant in Caucasian, that may not be predominant in the suppose Chinese population. So will the drug work in them, and what should we do in that case? And those are the kind of things that I think, like, at a very basic level, that we see that importance. And also, like, the trial inclusion and trial representation of the subgroups, that's also very important in terms of efficacy or effectiveness. Not efficacy, but I would say more on effectiveness, that whether that will be effective in that population or not, because we will not get the representative population in our trials. So
Got it. You're too humble.
no, I'm not going to go, and there are other experts. I think they'll be more helpful in those areas.
But again, thank you very much for joining us today. You've been just fascinating to have this conversation, especially exploring an area that we haven't explored too much in, which is really using real world data and real world evidence in the R&D space. So thank you very much for at least giving us a little bit of a glimpse into your world and into obviously in California right now, and a glimpse into California as opposed to us in Canada, which we're freezing ourselves off right now. But Costas, I don't know if you have anything else you would ask Shahed.
No, I again want to thank you for the insights. And I've got to say, we share your pain with a quality question. It is certainly something that bedevils every researcher. For us, it's always core in what we do and trying to pull together data that's useful for research. But we're itching to see how we can actually push that envelope with the use of real world data for R&D purposes. So
Yeah,
thanks again.
Thank you for inviting me to this podcast. And I do share the view that I think real world evidence will be very, very important throughout the product life cycle, from inception to the end. So thank you.
Excellent. Thank you very much, Shahed. So if you want to see more of Shahed's work, you can find him on Shahed Iqbal on LinkedIn. So is that the place that people can probably connect you with you easiest?
Yes, that's the professional platform I'm on.
Perfect. Do you do Twitter or X or?
No, I'm just on LinkedIn, and I think, like, that's where I see meet professionally with people, and I'm happy to connect with a lot of people and discuss. Always there for a good discussion.
Great. Thanks a lot. So thank you very much for joining us today, and thank you everyone for listening in today. And if you, of course, have any questions for us, please comment below, like and subscribe. And again, thank you again for joining us on another episode of What's with RWE. Thank you, everyone. Have a great day.
Thank you.
Bye, everyone.